Transcribe so hard scientists want to find me. First they gotta sequence me. What's the process to a cell like me? Can you please remind me? Well, it's foundation of a science and a rule of thumb. A series of processes bringing life with essential dogma as their sum.
Just some biomolecules encoding all the life we know, switching from one to the next in a nice, neat flow. DNA to RNA to proteins in the cell, a universal constant as far as we can tell.
But wait, if only it were actually that simple. We have this thing named HIV that's worse than a pimple.
It uses a protein called reverse transcriptase that throws the central dogma flat on its face.
We all know that AIDS is that nasty disease about the immune system.
We also know that AIDS is caused by HIV.
HIV belongs to a family of viruses known as the retroviruses.
They only possess RNA, which makes them very unique, which you would think makes their outlook seem very bleak. How do they produce? How do they persist? I don't know!
But wait, there's more. This thing called reverse transcriptase. Reverse transcriptase is a speed bump for the central dogma. Creates DNA from RNA, the answer to all our questions.
Now you can ask what creates mRNA. Lucky for you, that's now what we're going to say. Doesn't come from nowhere, it comes from inside the nucleus where the DNA resides.
First we have the sigma factors binding the polymerase, altogether binding to the promoter DNA strand. This creates a transcription bubble separating the strands. RNA polymerase now adding the nucleotides. We have ourselves a growing mRNA strand.
Now sugar phosphate backbone forms the H bonds break in two. A strand is now complete and is free to leave the DNA. If a cell has a nucleus, it may be further processed. Polyadenylation capping splicing is the hottest. Without a nucleus, these are prokaryotes. Transcription translation concurrently happening.
In bacteria, genes are organized into operons. A single gene promoter is needed to turn all of them on. In eukaryotes, it's not so simple. The action's in the spliceosome, with exons getting covalently joined and introns cut right out. This gives a boost to fitness, more variety in genes, allowing the encoding of even more proteins.
Now that we have shown how to create all this mRNA, let's use the northern blot to study the mRNA. While you may have heard of a southern blot for DNA, that's not the same as a northern blot for mRNA.
We can use a northern blot to study how much a gene is expressed, a little or a lot. This technique extracts RNA from an agarose gel onto DBM paper where it can be hybridized at swell. Now we will go into more detail, and the whole process we will unveil.
Step 1. Extract all the mRNA from the cell with protease. Step 2. Isolate and purify the mRNA. Step 3. Separate the sample by size using electrophoresis. Step 4 is a key innovation in the northern blot, the transfer of RNA bands onto paper called blotting.
It's not so easy because RNA doesn't bind to nitrocellulose. What they did was develop DBM paper. DBM is indiazobenzyloxymethyl. RNA covalently binds to this paper and can be removed. Of this past step, they greatly improved, after which can be hybridized with a radioactive single-stranded RNA probe to detect certain sequences.
Northern blot works well for genes that are highly expressed. It does not work for genes with lower expression rates. Instead, reverse transcription polymerase chain reaction works with high sensitivity. First, take the RNA, make it into cDNA. Next, amplify it using PCR. Now we can run it through a gel to analyze it.
Talked about bacteria, eukarya a bit, but there's a third domain of life just as important as the rest. And what's surprising are the similarities between them, as if RK has conserved an aspect of transcription from the past.
RNA polymerase gave us the first clue, with at least 10 subunits, quite a bit more than a few. Much more complex than bacteria, but still not quite brand new, just like eukaryote homologs that we already knew.
That was from initiation, but not elongation, with NUSA and NUSP factors from bacteria being found, controls for elongation and modulate read-through. In addition to this, they provide some anti-termination.
Holy cow, wonder what this could mean. Common ancestor connecting the three, quite possibly.
With gene regulators like those from bacteria also found, the theory's looking sound.
But we're still not there, not done.
But hey, it's an intriguing thought.